Module 8, Other Facultative and/or Glucose Non-Fermenting Gram-Negative Rods. As usual, I give you a big list of organisms in the beginning that I'm not going to read to you, even though you see some very interesting names in here. I'll read them to you as we go through the lecture. But there was a term that we used, facultative. And just a definition, a facultative anaerobe is an organism that makes ATP by aerobic respiration if oxygen is present, but is capable of switching to fermentation if oxygen is not there. So what are some of the general characteristics? Well, they're all gram-negative rods. And beyond that, each chapter groups together the most similar characteristics. For example, they're oxidase, their reactions can vary, they have varied growth on McConkey. Most are environmental organisms, mean that they're found in the soil and around places where, you might be outside or, pretty much anywhere. And they do have certain types of glucose utilization. There is important distinctions based on the glucose utilization. We have glucose oxidizers that can utilize glucose, but not by fermentation. Those are called saccharolytic. Glucose non-utilizers that do not oxidize or ferment glucose are called asaccharolytic. Many of the same media and biochemical tests discussed in Module 7 will apply here, but we'll add a few as necessary. The first organism is Acinetobacter. This is another genus that has undergone some major name changes. They've done away with the species names and they've just divided it into 4 major groups. There's a saccharolytic non-hemolytic, which is Acinetobacter a baumannii, a saccharolytic hemolytic, asaccharolytic non-hemolytic, which is Acinetobacter wolffii, and asaccharolytic hemolytic. These are similar to the Enterobacteriaceae, where they're oxidase negative. Most of them will grow well on McConkey. They're lactose non-fermenters, which means they're a clear colony, where the lactose fermenters were a pink colony before. They're different from the Enterobacteriaceae in that they are glucose non-fermenters that are either saccharolytic or asaccharolytic. They're a very important cause of nosocomial infections that you'd get in the hospital. And it can involve genital urinary tract infections, respiratory tract, wounds, soft tissues, and bacteremia. It's found widely distributed in nature. We find it in hospital environments, and they can colonize patients that are hospitalized for very long periods of time. The mode of transmission is indirect. It's usually from some type of IV or catheter. It doesn't really have any known virulence factors, but there is antibiotic resistance to carbapenems. This contributes to the severity of the infections where we find it. On blood agar, it is smooth, opaque, raised, creamy, smaller than Enterobacteriaceae, and some are hemolytic. On McConkey, it's a non-lactose fermenter, but the colonies may have a purplish hue that can be mistaken for a lactose fermenter. Some other biochemical tests, it's oxidase negative, motility negative. It is saccharolytic, which means it's positive for glucose utilization. It's asaccharolytic or negative for glucose oxidization. Oxidization. Boy, that's a tongue twister for me today. This one is saccharolytic. It has variable maltose utilization. It's esculin hydrase negative, lysine decarboxylase negative, nitrate negative, and urea variable. It is resistant to some beta-lactams, carbapenems, aminoglycosides, and quinolones. Susceptibility testing can be done with disc diffusion, broth dilution, or agar dilution. Here's the esculin hydrolysis test. This is a presumptive identification for various Enterobacteriaceae and some other organisms. The principle of this is to determine whether an organism is capable of hydrolyzing the glycoside esculin. Esculin is hydrolyzed to esculitin, which reacts with iron and forms a dark brown-black precipitate. It's positive if we see blackened media with a loss of fluorescence under a Woods lamp. It's negative or no blackening and no loss of fluorescence under the Woods lamp. We also look at oxidation and fermentation of the medium. This test, the OFB test, is for differentiation of microorganisms based on their ability to oxidize or ferment specific carbohydrates. So how we do this is the carbohydrate of interest is added to the media. 2 tubes are inoculated, one with mineral oil as an overlay to achieve an anaerobic environment. The production of acid in an anaerobic tube indicates fermentation. Production of acid in an open tube is the result of oxidation. The next organism is Stenotrophomonas multifida. Most of these infections are nosocomial. They can be catheter-related, can cause bacteremia, wound infections, pneumonia, and UTIs. It's widely distributed in nature. We can find it in hospital environments. It can become normal flora of long-term hospitalized patients. The mode of transmission is usually indirect, such as colonization due to hospitalization or medical instrumentation. We don't know much about the virulence factors, but it does grow in common media. On a blood agar, there's large, smooth, glistening colonies with uneven edges and lavender green to light purple pigment, a greenish discoloration underneath, and it smells of ammonia. On McConkey, it'll grow, but it's a clear and colorless colony called a non-lactose fermenter. Other tests, it's oxidase negative, motility positive. You can kind of see by how it kind of smeared over the plate here. It is a glucose oxidizer, so it's saccharolytic. It's a maltose oxidizer. The esculin hydrolysis is variable. The lysine decarboxylase is positive. Its nitrate reduction variable and urea negative. As far as antimicrobial susceptibility testing goes, it's intrinsically resistant to most beta-lactams and aminoglycosides. The next organism is Burkholderia gladioli. This has an unknown or uncertain role in human disease. Occasionally we'll find this in the sputum of cystic fibrosis patients. Its habitat is environmental, usually in plants. It's not considered normal flora even when we find it in cystic fibrosis patients. The mode of transmission is unknown, and the virulence factors are unknown. But the appearance on common media, blood agar, we see yellow colonies. McConkey, non-lactose fermenter, clear and colorless. Other tests, oxidase is negative, motility positive. It's saccharolytic. It's a maltose oxidizer. Esculin hydrolysis is negative, lysine decarboxylase negative, nitrates variable, and urea is variable. As far as susceptibility testing goes, the potential choices include imipenem, piperacillin, and ciprofloxacin. This next one is very important in our cystic fibrosis patients. It's called the Burkholderia cepacia complex. This is able to colonize and cause life-threatening infections in cystic fibrosis patients or patients with chronic granulomatous disease. Other infection may include UTIs, respiratory tract, or sterile body sites. The species is the most common Burkholderia species in clinical specimens. It's widely distributed in nature. We find it in hospital environments, and it's a colonizer of the respiratory tract of those cystic fibrosis patients. Now, in the course agenda, I did post a trailer to a movie called 6 Feet Apart. The 6 feet apart, or is it 5 feet apart? 5 feet apart? 6 feet apart. Either way, these cystic fibrosis patients in the movie are required to keep a certain distance apart from each other. This is very, very contagious for cystic fibrosis patients, and once they get it, it can be a death sentence. The mode of transmission is usually indirect or by having exposure to medical devices, but it can be transferred from cystic fibrosis patient to cystic fibrosis patient, so they usually are required to keep away from each other. It has virulence factors because it can bind the mucin from cystic fibrosis patients. Cystic fibrosis patients have a lot of excess mucus in their lungs and in their intestines and all different areas of their body. And because it has such an affinity for binding to mucin, it causes a lot of problems. It has a lot of antibiotic resistance, which makes it very difficult to treat. On blood agar, it has a smooth and slightly raised or dirt-like odor. On McConkey, there's non-lactose fermenters where the colonies become dark pink to red because of oxidation of lactose after 4 to 7 days. On chocolate, it creates kind of a green pigment. There is other media. There's Burkholderia cepacia selective agar, and there's an oxidative fermentive base polymyxin B bacitracin lactose agar, which is also used for this. OFPBL agar is easier to say. So here's some Burkholderia cepacia selective agar. It's for the recovery of just B cepacia. It's got bile salts, gentamicin, ticracillin, polymyxin B, peptone, and yeast agar. It inhibits the growth of most organisms, but it does allow the growth of B cepacia. The OFPBL agar is also for the recovery of B cepacia. It contains polymyxin B and bacitracin. It also contains lactose to differentiate from those that can ferment or not. Organisms that can ferment lactose appear yellow, and organisms that cannot appear green. The other biochemical test that we can do with B cepacia includes oxidase, that's positive. It is a glucose oxidizer. It grows at 42 degrees. It's variable for nitrate reduction and gelatin liquefaction. It's arginine dihydrolase negative, lysine decarboxylase positive, urea variable, and lactose oxidizer positive. The antimicrobial susceptibility testing potential choices include SXT, timentin, ceftazidime, and merenipin. Antimicrobial therapy rarely eradicates the microorganism completely, but the development of resistance can occur. This next one is Burkholderia mallei. Human disease is very rare, and infections range from skin infections at the inoculation site to acute pulmonary infections and septicemia. This can cause something called glanders in horses, mules, and donkeys. Therefore, the reservoir is horses, mules, and donkeys, and it's not a normal human flora. The mode of direction would be having close contact with animals, and it can be introduced through the mucous membranes and broken skin. There's no real distinctive appearance on blood agar, and it's a non-lactose fermenter on a McConkey plate. It is oxidase positive. It's saccharolytic. It won't grow at 42 like cepacia will. Nitrate reduction positive, gelatin liquefaction negative, arginine dihydrolase positive, lysine decarboxylase positive, urea variable, and lactose oxidizer positive. Resistance can occur, and potential choices include ceftazidime, beta-lactams, and clavulanic acid. Next, we have Burkholderia pseudomallei. This ranges from asymptomatic infections to something called melioidosis. Melioidosis is a skin or respiratory tract abscess. It affects the skin and respiratory tract, causes abscesses, bacteremia, and septic shock. We find this one more in tropical regions, primarily Southeast Asia. It's not normal flora to humans. They can get it from the inhalation or inoculation from the environment. It can survive phagocytosis, which is a virulence factor. On blood agar, it's cream to yellowish orange, smooth and mucoid to dry and wrinkled, and has a putrid odor. On McConkey, it's a non-lactose fermenter, but it can appear pink as it can oxidize lactose. Other media, ashdown agar. It causes a dry, wrinkled, violet-purple colony. It's kind of cool looking. So the ashdown agar is for the isolation of Burkholderia pseudomallei. It contains crystal violet and gentamicin for the inhibition of most organisms, and it's got glycerol for the growth of B pseudomallei. Neutral red acts as a color indicator. So as you can see on here, B pseudomallei will grow and absorb neutral red and become dry, wrinkled, in a violet color. Other biochemical tests, it's oxidase positive. It's a glucose oxidizer. It does grow at 42 degrees. It's nitrate reduction positive, gelatin liquefaction variable, arginine dihydrolase positive, lysine decarboxylase negative, urea variable, and lactose oxidizer positive. With this one, antimicrobial susceptibility testing can be done and prolonged therapy may be required because there is a potential for resistance. The potential choices include ceftazidime, beta-lactams with clavulanic acid, meropenem, SXT, and doxycycline. This next one is very common. So this one is called Pseudomonas aeruginosa, and it's one of my favorites. So it's most common next to the Enterobacteriaceae. It's an opportunistic pathogen. We find it in community or hospital-acquired infections. We see it in external ear canal infections. If you've ever had swimmer's ear, it's a common thing for that. Infections can follow trauma, bone, heart, and eye damage. Can be found in IV drug users. Can be found in the respiratory tract of those cystic fibrosis patients we just talked about. It can cause UTIs, wound bacteremia, and central nervous system issues, and it's a problem with burn wounds as well. This guy is found in the environment, soil, water, and plants. Think of like a pond or stagnant water. If a hot tub, whirlpool, contact lens solution is let sit too long, Pseudomonas aeruginosa can grow. We can find it in sinks, showers, and respiratory equipment as well. It's rarely part of normal human flora. We usually get it from the ingestion of contaminated food or water or exposure to contaminated medical equipment. Person-to-person transmission can occur, although it's probably a lot less likely than the environmental transmission. This guy has some serious virulence factors that we do know about. It has exotoxin A that inhibits protein synthesis, exoenzymes S and T that disrupt cytoskeleton organization. It's got a lipopolysaccharide endotoxin, proteolytic enzymes and hemolysins, pili and adhesions. It's got intrinsic resistance to many antimicrobials, and it has alginate. Alginate is a polysaccharide polymer that inhibits phagocytosis and contributes to antimicrobial resistance, and it protects the organism from dehydration. On blood agar, it's spreading and flat with serrated edges, has confluent growth, often has a metallic sheen. It's kind of a bluish green color, but it can be red or brown pigment. It has a beta hemolytic type of look to it, and it has a grape-like odor. Mucoid colonies are seen in cystic fibrosis patients. On McConkey, it's a non-lactose fermenter. It grows clear and colorless. Occasionally, you may see a bit of a greenish pigment. On Mueller-Hinton agar, it's got a bright bluish green pigment. That plate is shown on the next slide. Sorry, here we are in the next slide, the slide after this. This one is the blood agar plate. So this one, it's oxidase positive. It's saccharolytic, grows at 42 degrees. It has alkaline over no change on TSI media, which is what we used for the Enterobacteriaceae. It's nitrate reduction positive, gelatin liquefaction variable, arginine dihydrolase positive, lysine decarboxylase positive, and urea variable, and it is a negative lactose oxidizer. You can see it's kind of a flat growth, kind of a little grayish greenish in the back there. Here is the agar I was just talking about, the Mueller-Hinton. So with the antimicrobial susceptibility testing, it's intrinsically resistant to many antimicrobials. It has a potential for resistance to develop during treatment. Potential choices include beta-lactams with an aminoglycoside. Some quinolones may be used as well. We do have other Pseudomonas species such as fluorescens, putida, stutzeri, and these are an uncommon cause of infections, but if they do cause an infection, it would be bacteremia, a UTI, a wound, respiratory tract, or central spinal fluid. The significance in culture should be questioned if we do see them. These are found in soil and water. They're not considered normal flora. We can get it from contaminated medical devices or solutions. Its virulence factor is unknown, and it can cause infections in immunocompromised patients. In blood agar, there's no distinctive appearance. In McConkey, it's clear and colorless. Other biochemical tests, it's very similar to Pseudomonas aeruginosa. And antimicrobial susceptibility testing, there's no validated testing methods. And the treatments that we use for regular Pseudomonas aeruginosa are pretty effective for the other ones. An interesting side note, Pseudomonas aeruginosa, Pseudomonas fluorescens, and a few others produce pyoverdin, which fluoresces under a UV light. The next one is Ralstonia pickettii. This rarely causes disease, and it's non-pathogenic in healthy humans. Generally, it's considered a contaminant when isolated in clinical specimens. This is found in soil, water, and sludge. It has an unknown mode of transmission. Its virulence factors are unknown. But on blood agar, it has no distinctive appearance, but it can take up to 72 hours to produce colonies. On McConkey, they have clear and colorless colonies. All right, next one is Aeromonas. Aeromonas caviae complex and Aeromonas hydrophilia complex are the most commonly encountered. This causes gastroenteritis, wound infections, and bacteremia. Sometimes we see it with endocarditis, meningitis, pneumonia, conjunctivitis, and osteomyelitis. In the environment, we find it in the aquatic areas, freshwater, polluted or chlorinated water, brackish water, which is kind of a salt and freshwater mix, and occasionally in marine water. It may transiently colonize the GI tracts of various animals. We can have an indirect transmission, which would be from eating seafood, or indirect, which would be exposure to disrupted skin. Could be from like a fish fin, like that pokes you, or a fish hook. It does have some various toxins, but the specific role is uncertain. With Aeromonas on blood agar, there's a large, round, raised, opaque, mostly pathogenic strain that are beta hemolytic, except for Aeromonas caviae. On McConkey, it can either be a non-lactose fermenter or lactose fermenter, where they can be clear, colorless, or pink. They're oxidase positive. They can ferment and oxidize glucose. They're usually indole positive, and they do not grow in 6% sodium chloride. As far as antimicrobial susceptibility testing goes, they are capable of producing beta-lactamase. And usually gastritis is self-limiting, which means it goes away on its own. The next one is Vibrio cholerae. This causes cholera, which is a profuse watery diarrhea leading to dehydration, hypotension, and often death. We find this in epidemics or pandemics of Vibrio cholerae. It can cause non-epidemic diarrhea, occasionally infections of wounds. We see it in the respiratory tract, urinary tract, and the central nervous system. Cholera toxin causes a profuse watery stool, also known as rice water stool. It leads to dramatic fluid loss, severe dehydration, and hypotension that often leads to death. We find this one outside of the human GI tract between epidemics and pandemics. It may survive in dormant state in brackish or salt water. Human carriers are also known, which are particular in the endemic regions. The mode of transmission is fecal oral, so ingestion of contaminated washing, swimming, or cooking water or drinking it. Also by the ingestion of contaminated shellfish or seafood. It does have that cholera toxin, which causes hypersecretion of fluids and electrolytes into the lumen of the GI tract. There's also Zonula occludens toxin, or ZOT, which is an enterotoxin. It has ACE, which is another enterotoxin. It has some other cytotoxins, and it causes a lot of issues in those intestines. On common media, blood agar, it has a medium to large, smooth, opaque, iridescent with a greenish hue. It's usually beta hemolytic. Non-lactose fermenter. Other selective media, TCBS agar, which is used for the differentiation of Vibrio species or Vibrio cholera. And other biochemical tests, we can do a string test, which is used to differentiate Vibrio from Aeromonas and Plesiomonas. That's important to know. They are oxidase and indole positive. They can ferment glucose, and they do grow in 6% sodium chloride. As far as antimicrobial susceptibility testing goes, first thing they will do is replace the fluids by giving IVs. It's resistant to tetracycline and chloramphenicol and SXT. Potential choices include aminoglycosides, fluoroquinones, and extended spectrum cephalosporins. On the TCBS agar, it stands for thiosulfate citrate biosalt sucrose. This is for the differentiation of Vibrio species. We have a peptone base agar with yeast extract, biosalts, and sodium thiosulfate for inhibiting most organisms. Citrate, sucrose, and ferric citrate for the differentiation of Vibrio. And it has bromthiamyl blue as a color indicator. Yellow colonies indicate sucrose fermentation and green is non-sucrose. As far as the string test goes, this differentiates Vibrio species from Aeromonas species and Plesiomonas. Organisms are emulsified in a 0.5% sodium deoxycholate which lyses Vibrio cells. The cell lysis releases DNA that can be pulled up in a string. If it's positive, it's Vibrio species. Negative, Aeromonas or Plesiomonas gigaloides. The next one is Vibrio vulnificus. This can be found in wound infections and septicemia. Usually not gastroenteritis, but it can. We find this in brackish or saltwater, which means it's halophilic. Usually it's from the ingestion of contaminated water or seafood. On blood agar, they cause medium to large, smooth, opaque, iridescent with a green hue, and usually non-hemolytic. On McConkey, they are lactose fermenters, so they're pink, and TCBS, they cause green colonies. In other biochemical tests, they're oxidase positive, indole positive, they can ferment glucose, they grow in a 6% sodium chloride positive, and their string test is positive. As far as antimicrobial susceptibility testing goes, there's known resistance to tetracycline, chloramphenicol, and SXT, and there's no definitive guidelines. They usually use the same for Vibrio cholerae, same treatment. The next one is Campylobacter jejuni. This is commonly causes gastroenteritis, but it can cause septicemia, meningitis, and actually proctitis as well. It is a normal intestinal flora of pigs, poultry, bulls, dogs, cats, birds, and other animals. We get this from the contamination of food, milk, or water with animal feces. In developed countries, it's usually indirect from improper food preparation. Indirect is from person-to-person transmission. It has some virulence factors, and it's kind of like a cholera toxin-like toxin. Campylobacter likes to grow in 42 degrees. We can also use a Campy-CVA agar, which is a blood agar with supplements that require 48 to 72 hours for growth. On the Campy-CVA agar, it's a Brucella agar base with a cephalosporin, vancomycin, and amphotericin B, and 5% sheep's blood agar. The growth of colonies in microaerophilic conditions in 42 degrees should be gram stained and oxidase tested for the identification of Campylobacter. Campylobacter has a famous gram stain known as gull wings. They're gram-negative rods that look like seagull wings. They're oxidase positive, hippurate positive, catalase positive. There's no growth at 25 degrees, and it does grow at 42 degrees. Erythromycin is the drug of choice, and there is some resistance developing to macrolides and fluoroquinolones. One of our last organisms is Helicobacter pylori. This one causes gastric ulcers. The primary reservoir is the human gastric mucosa. The mode of transmission is unclear, but it's usually oral to oral, fecal oral, environmental, or mother to child transmission. It's known for its urease production. It hydrolyzes urea, forming ammonia, which increases the pH and turns it pink. The ability to invade the gastric epithelium is mediated by something called a CAG-A protein. Cultures are not routinely done, but instead we do serodiagnosis. This is a detection in gastric biopsy. It's a spiral-shaped bacteria, and H. pylori breath test. We can also do antigen antibody testing and PCR testing. With antimicrobial susceptibility testing, in vitro tests can be unreliable. Resistance develops relatively easily only when one agent is used. Current regimens recommend triple drug therapy and a proton pump inhibitor for temporary relief of infection. And here you can see it inside the stomach mucosa. All right, here we have some case studies. I'm going to include the answers to these case studies in an additional document below the PowerPoint today.